Welcome back to The Explainer. Today, we're putting on our detective hats to solve the ultimate biological mystery. How exactly are traits passed down? We're focusing strictly on Chapter 7, Heredity, and we're going to dig right into those biological blueprints that make us, well, us.
All right, here's our roadmap for Chapter 7. We'll start with the language of traits, move beyond simple dominance, hit Mendel and genetic crosses, map out some gene linkage, decode pedigree charts, and finally wrap up with epigenetics. Let's get to it.
So before we start examining the hard evidence, think about this for a second. Why do some traits skip a generation entirely while others seem to just appear out of absolutely nowhere? We've all wondered this when looking at our own families, right? Well, the answer lies hidden in the biological clues of heredity.
Section 1. The language of traits. Building our detective toolbox.
Okay, so the absolutely crucial starting point is knowing the difference between the genotype, that invisible genetic blueprint locked away inside our cells, and the phenotype, which is the physical trait we actually see. Think of the genotype as the hidden source code of a mobile app, and the phenotype, that's the final user interface you interact with, like your height, your blood type, or the color of your hair.
And what actually makes up that genotype? Alleles. You can think of an allele as a specific flavor or a variation of a gene. We get one from each parent, and they sit at the exact same locus, basically their physical street address, on our matching chromosomes. Now, these alleles can be dominant, actively masking the others, or they can be recessive, which means they only show up in the phenotype if there's no dominant allele around to steal the spotlight. Now, looking at things like eye color really brings home two vital concepts: penetrance and expressivity. Penetrance is just the proportion of people who actually show the phenotype for a specific allele. Like, not everyone with a certain disease gene actually develops the condition. And then there's expressivity. Even if two people have the exact same genotype for a trait, let's say curly hair, the actual physical expression, the degree of curliness, can completely vary from person to person. So it's not always just a simple on and off switch.
Section 2. Beyond simple dominance. The rule breakers.
Check out these beautiful hibiscus flowers. On one hand, we've got incomplete dominance creating a gorgeous intermediate pink flower from a red and white mix. Basically, neither allele completely wins out. But then, look at codominance. That's where both the red and the white alleles fiercely share the spotlight, expressing both colors at the exact same time as brilliant red and white spots.
And then there's a wild phenomenon called epistasis, where one entirely different gene completely masks the expression of another. It's a real game changer. It perfectly explains why two dark-coated Labradors can have an adorable golden puppy. The light coat gene essentially steps in and just overrides the genetic instructions for black or chocolate fur. Nature is crazy like that.
Section 3. Mendel and genetic crosses. The foundational rules.
When we're solving a genetic cross to figure out how traits get passed down, we follow a strict chronological sequence. We start at the top with the true breeding P-parent generation, trace down through their children, the F1 hybrids, and finally to their grandchildren, the F2 generation. It's all about predicting those traits down the family line.
So, looking at a single gene in a monohybrid cross, Mendel's law of dominance really comes to life. By crossing two F1 hybrids, we reveal a beautifully predictable, classic 3-to-1 ratio of dominant to recessive traits in the F2 generation. You get 3 showing the dominant phenotype and exactly 1 showing the recessive.
But what happens when we examine two completely separate genes on separate chromosomes in what we call a dihybrid cross? Well, the math expands. We get this beautifully complex ratio of 9 to 3 to 3 to 1 for the resulting phenotypes. That's 9 dominant for both traits, 3 mixed one way, 3 mixed the other, and just 1 recessive for everything. The whole shebang in one elegant ratio. Section 4. Mapping gene linkage. Connecting the clues.
If genes are tightly linked together on the same chromosome, they actually defy Mendel's independent assortment. They tend to travel as a package deal. But, as we know, nature absolutely loves variation. Crossing over during cell division can occasionally separate these linked genes to create unique, recombinant gametes with brand new allele combinations.
We can actually measure the physical distance between these linked genes using map units. And the formula is beautifully simple. One map unit perfectly equals a 1% chance of recombination between two genes per generation. So, to put that into perspective, a distance of 20 map units means there's a 20% probability those genes will be split up by crossing over.
Section 5. Decoding pedigree charts. Tracking the evidence.
Now this is where you can really test your detective logic. In a pedigree, squares are males, circles are females, and shaded means they show the trait. By looking at an autosomal dominant trait, we can deduce that an affected male in the third generation must absolutely be heterozygous. Why? Because there's no way he could have inherited a dominant affected allele from an unaffected father. Therefore, he had to receive a recessive allele from dad and the dominant one from mom. It's like a giant biological puzzle.
Section 6. Epigenetics. Regulating the code.
Finally, we hit the massive plot twist of Chapter 7. Epigenetics shows us that by modifying DNA with molecules like methylation or acetylation, the exact same genetic code can be expressed completely differently without ever mutating or changing the actual DNA sequence. It acts as a set of physical switches deciding when and how strongly a gene is turned on or off based on environmental factors like our diet or stress levels.
It really makes you wonder just how adaptable our blueprints are, doesn't it? If epigenetics can change how our code is expressed without altering the DNA itself, how much of our genetic destiny is truly set in stone? That leaves us with a totally fascinating mystery to ponder long after this explainer ends. Keep being curious and keep decoding the world around you.